All right, today we are going to be covering chapter 4, which is over cellular metabolism. All right, again, as always, we're starting out with some roots, prefixes, and suffixes. AER, aerobic, like think of aerobics. That's something that requires oxygen. AN, without. We'll talk about anaerobic respiration, things that don't require oxygen. ANA versus CATA, anabolism versus catabolism. All right, so we know cells work 24/7, 365. We know that, well, we don't know. Chemical reactions have to occur inside the body in a precise reaction. We know, or sometimes, chemical reactions, when we do them, we don't necessarily in the body just stop with A plus B equals AB or A plus B yields AB. But in the body, usually what happens is that we have, 157 steps before we get to our final destination. So A plus B equals AB plus C plus D plus E plus F. And then finally we get to A, B, C, D, E, F. And that's what the body needs in order to create a protein called A, B, C, D, E, F. And I'm just making that up. In reality, the protein might be A, B, C, D, E, F, G, H, I, J, K, L, M, N, O, P, blah, 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 blah. And it might be, remember, amino acids can be, 5,000, or proteins can be made up of 5,000 plus amino acids. So we might have to do this process over 5,000 times. And with any addition or with any adding on of these reactants, we have to use energy. We have to use ATP in order to get these reactions to go. And in order to maintain homeostasis, cells can't have too much or too little energy. So we have to make energy as we go. Energy is made in mitochondria. Remember that DNA carries the instructions to synthesize that protein, to synthesize those proteins, and RNA carries out those genetic instructions. Body uses the fats, carbohydrates, and proteins to accomplish cellular metabolism. And I've already said that carbohydrates are our first line of defense for generating ATP, for metabolizing glucose. Carbohydrates get processed down to glucose to generate ATP, which we'll talk about in depth shortly. All right, so metabolism. What is metabolism? Metabolism is a sum of all chemical reactions that are occurring inside the body. We have cellular metabolism. Typically, they occur in either pathways or cycles. A pathway is a stepwise process. When we look at glycolysis, we have step 1, step 2, step 3, step 4, step 5. And once we get to the end, we're done. A cycle is something that looks like a circle. Citric acid cycle occurs in a circle where we kind of have a beginning and an end, but the end products will kind of roll over into the beginning. And as long as we have enough end products and as long as we have enough source material coming in, the cycle will continue and continue until we don't have any more end products, we don't have any more catalysts or enzymes, or we don't have any more source material. But we'll get to that later on. All right, so we have two different types of overall generic metabolic reactions: anabolism and catabolism. Anabolism, we actually talked about these in the chemistry chapter in chapter 2, but we called them a little bit different things. Synthesis reactions and degradation reactions. Remember, synthesis reactions were reactions where we add things together, and degradation reactions were where we broke things apart. That's what anabolism and catabolism are. Anabolism are where we take smaller molecules and we build them into larger ones. Remember we have that glycerol plus the 3 fatty acid side chains make a triglyceride molecule. Or we combine monosaccharides to form glycogen. Remember I mentioned that glycogen is just a bunch of glucose molecules that are stored in your liver and skeletal muscle cells to be used for energy later on. Catabolism is a degradation reaction. It's where we take larger molecules and we break them down into smaller ones. Some examples, we break down that dipeptide into 2 amino acids. Remember, amino acids are joined together by that peptide bond. So we have a dipeptide, 2 amino acids. We break them down into each subsequent amino acid. Trypsin, that's an enzyme found in the small intestine that breaks down proteins into amino acids or into smaller ones. Amylase, we actually have amylase in our saliva and our pancreas, salivary amylase and pancreatic amylase. That's going to break down starch into either disaccharides or monosaccharides. We're breaking down something big, starch, into something smaller. Breaking down glucose in the cytoplasm and mitochondria. That's going to be what we spend a lot of this chapter discussing. Glycolysis, citric acid cycle, electron transport chain, pyruvate oxidation, breaking down glucose in the cytoplasm and mitochondria. All right, anabolic reactions, adding things together. Smaller molecules are bound together to form larger ones. Synthesis reactions. These are biochemical reactions that synthesize compounds. Typically, we use this for cellular growth and repair. Because we are building up something, these reactions require ATP, and we use that ATP, or that ATP that we use comes from catabolic reactions. It's made during catabolic reactions. We call these dehydration synthesis reactions. So, for example, we can use to produce disaccharides, polysaccharides, proteins, triglycerides, nucleic acids. We're building something up. And it says we're producing water, or water is released in the process. And it says, an OH. This statement's probably going to be a little confusing. Right here, an OH from the COH group, 1 amino acid, blah, blah, blah. So we'll come back to this statement here in a second. All right, catabolic reactions. We're breaking something down in a large molecule in a smaller one. Remember, those are our degradation reactions. Chemical reactions that release energy. So in this case, ATP is produced. Now, when we produce ATP, 60% of that ATP is generated as heat, and 40% of that ATP is used, or 40% of that energy is used to rebuild molecules, transport substances, cellular repair, structural repair. So examples, glycolysis, pyruvate oxidation, Krebs cycle, citric acid cycle. Sometimes we'll call it citric acid cycle, sometimes we'll call it the Krebs cycle. Electron transport chain. We're breaking down substances. We also call these hydrolysis reactions. Basically, it's the reverse of dehydration synthesis. Breaking down substances. We are decomposing or breaking down carbs, proteins, and lipids. So, for example, digestion. Digestion, we break down something. In this case, we're going to use water to split the big molecule into 2 smaller molecules. And we'll, like I said, a little confusing right now. We'll see examples of this here in a second. So sucrose, which is a 2-sugar molecule, is going to be broken up to yield a glucose molecule and a fructose molecule. All right, so here in this example, we have a monosaccharide plus a monosaccharide to yield a disaccharide plus water. So we talked about producing water. So up here in anabolic reactions, water is produced or released in the process. Dehydration synthesis. We're taking two smaller molecules and we're making a bigger one. So technically we should probably put a plus there. So here what we're doing is we're taking an OH and an H from here, and that's how we get our water molecule here. And we're taking 2 molecules and we're now making 1 molecule. So you can see this double arrow right here. And remember that I said that 99% of the time and that's a reversible reaction, that 99% of the time our bodies are going to have those double-headed arrow reactions. And it depends on what our body needs are at the time. So if our body needs more disaccharides, we're going to go this way. If our body needs more monosaccharides, we're going to go this way. If we go from left to right, we're going to do the anabolic reaction or the dehydration synthesis reaction. If we need more monosaccharides, we're going to go from right to left. We're going to do the hydrolysis reaction or the catabolic reaction. And in that case, we're going to use water. We're going to put water right here, and we're going to split up this molecule into 2 monosaccharides. So in the dehydration synthesis, 2 monosaccharides combine to yield a disaccharide and water. So an example is we would eat pasta the night before a swim meet. Catabolism or hydrolysis would break down the starch, that polysaccharide, into glucose, which is a monosaccharide. The glucose molecules would be stored as glycogen in the liver and skeletal muscles via a process called glycogenesis. We'll talk about that later, which is a dehydration synthesis binding the glucose molecules together. Then during your swim meet, you would need that immediate energy. The cells would then break down, or that hydrolysis reaction, that glycogen via a process called glycogenolysis to release that glucose from that glycogen. All right, so then I said, this example right here, we'd come back to this later on. All right, so we have the amino acid example. Amino acid plus amino acid yields that dipeptide plus that water. We have our COOH group, so we're going to take an OH from that COOH. We have that NH2 group. We're going to take that H from that NH group to produce our water. We're going to join those 2 amino acids and form that peptide bond there. If we need to build a protein, we're going to go this way, dehydration synthesis. If we need amino acids, we're going to go this way and do a hydrolysis reaction. If we need to go hydrolysis, we're going to use our water molecule, throw it in there, and go from right to left. And remember that peptide bond, this peptide bond is formed via the dehydration synthesis reaction with the amino acids as substrates or reactants. All right, so there's proteins. Now fats. Remember, fats are those glycerol plus 3 fatty acid side chains. So we would do this reaction 3 times. We would take our OHs and our H's. 1, 2, 3. We would form 3 water molecules. Going this way, dehydration synthesis. We're adding together. Going this way, hydrolysis. If we need more triglycerides, we would go from left to right. If we need more glycerol or more fatty acid molecules, we would go from right to left. If we needed more, we would throw in water molecules there, there, and there, and get glycerol plus fatty acid molecules. All right, so all cells are going to perform catabolic and anabolic reactions. Remember, it depends on what our body needs are at that time. And again, like I said, one example is producing glycogen to store excess glucose. And that rate of catabolism and anabolism is carefully controlled. We have to have that balance. Any imbalances can harm the cell. We do have enzymes that control the rates of both the catabolic side and the anabolic side. And that those enzymes or those catalysts increase the reaction time. And we talked about carbonic anhydrase a couple chapters ago. All right, just as a review, enzymes, those are protein catalysts that are found within the body. They increase the rate of reaction. They're not consumed. They don't undergo any conformational change. They can be used again and again. Remember, enzymes are proteins. They're essential to our metabolism. That is why protein synthesis is very important. In our earlier example, when we had this one, we would have an enzyme here to catalyze that reaction. We would have a different enzyme here to catalyze that one. We would have a different enzyme here to catalyze that one, a different enzyme here to catalyze that one, and a different enzyme here to catalyze that one. So each enzyme is specific to a particular substrate. So the same enzyme is not going to work is only going to work on a specific substrate. And we'll see that the conformation or the shape of that protein is going to be what drives what type of enzyme can attach to it. We'll see a picture version of this here in a couple slides. All right, we know that we talked about activation energy and how enzymes lower that activation energy. Here's that activation energy. So we've already seen this slide in chapter 2. All right, so typically enzymes end with the suffix ase. Now, most of the time you can go with what the prefix is as to what they break down. Lipase, LIP means lipids, so lipase breaks down lipids. Protease splits up proteins. Now, sometimes you can't. Hydrolase doesn't break down water, but it actually adds water during hydrolysis reactions. Amylase splits up starch, which is amylose. We'll skip this one. Lactase splits up lactose. Maltase splits up maltose. Sucrase splits up sucrose. RNA polymerase doesn't split up RNA, but it actually builds up RNA. Remember, using DNA as a template. Catalase actually speeds up the breakdown of hydrogen peroxide. Acetylcholinesterase breaks down the neurotransmitter acetylcholine. Insecticides work by inhibiting acetylcholinesterase. They had these things called chlorpyrifos that inhibit acetylcholinesterase, and that insecticide actually binds to the active site on acetylcholinesterase to inhibit the activity of acetylcholinesterase. And what happens is acetylcholinesterase can't break down acetylcholine. So you have a buildup of acetylcholine, and that leads to this condition called cholinergic crisis. And that's what happens like when you spray the yard with Roundup and your dog goes outside and licks the Roundup and comes back inside and starts having all these symptoms, and the dog might get cramps or muscle weakness, or they might start twitching a little bit. You might get blurred vision, diarrhea, abdominal cramps, urination, vomiting, eyes start to water, peeing on himself, headache, insomnia, confusion, drowsiness, sweating, low heart rate, low blood pressure, slurred speech, convulsions, coma, respiratory depression. That's all called cholinergic crisis, and we'll revisit this later on. All right, so I mentioned that that proteins have that specific shape, and that enzyme needs that specific shape in order to kind of work. So what happens is that we have those reactants. The A plus B yields AB. So we call those reactants substrates. So in this case, we would have A plus B here, and this is our enzyme down here. Instead of now calling them A plus B as being reactants, we're going to call them substrates. So here the enzyme and the substrate combine. And those substrates are proteins, and those proteins have to have a specific shape. Remember, we have that tertiary or that secondary level of protein shape due to hydrogen bonding, and that tertiary shape due to chaperone proteins, that three-dimensional structure due to chaperone proteins. So proteins have that specific shape. And if they have the correct shape, those proteins can kind of fit into this enzyme. And once they fit into this enzyme, because they have that correct shape, we now have this enzyme substrate complex, or this ES complex. And once the enzyme fits into this, or once the substrates fit into this enzyme, and we have this ES complex, the two substrates kind of fuse together. And now we have that AB product. And that reaction occurs. That enzyme speeds up the reaction of combining A and B together. Those products are released. And remember the definition of an enzyme. It's unaltered, it's unchanged, and it doesn't get used up. We can reuse that enzyme over and over and over. So what happens is, remember how we have A, B, C, D, E, and F. Each enzyme or each substrate has a particular enzyme that goes along with it. And we see these pathways that our body needs in order to get its end product. And as I said before, our end product might be 5,000 amino acids. So this process might be over 5,000 amino acids long, and we might have 5,000 different enzymes. So it's very enzyme controlled series of reactions, hopefully leading to our final product. And each new substrate is the product of a previous reaction. So here we have A plus B yields A, B plus C. Then we have A, B, C plus D. Then here we have A, B, C, D plus E. And then A, B, and then hopefully A, B, C, D, and E would be our final product. And like I said, each substrate has a specific enzyme needed in order for that reaction to take place. Now, what we also see is that we have this thing called a rate limiting enzyme. And basically, it's a regulatory enzyme that catalyzes one step of the pathway, typically sets the rate for the entire reaction sequence. All right, so what does that mean? So let's go back up here. So let's say that we have a classroom full of people, and I start out, and it takes me 5 minutes to do my job, and I have A plus B. And I put A plus B together, and then I give it to the next person, and they do they have A and B. Now they add C, and they give it to the next person. They add D, and they add E, and they add F, and they add G, and they add H, and I, J, K. And we go around the classroom until everyone adds their piece of the puzzle to the equation, and we finally have our end product. And if we do this for a long time, if we do this for days, eventually we'll have 1,000 different products, and we can say that on average it takes us about 5 minutes. It takes us 5 minutes to create one product, even though the first time that we do it, you guys are kind of waiting on me and waiting on everyone else for the first product and that second product to work its way around the room. But once we're all managing the assembly line, and once we all get going, on average, it takes us about 5 minutes to do our job, or 5 minutes to create one product. Now we throw in this thing called a rate limiting enzyme. And it says oftentimes the first enzyme in the reaction sequence is that rate limiting enzyme. So now let's say it takes me 10 minutes to do my job, but it still takes all of you guys 5 minutes to do your job. Well, now the average time it takes all of us to do our job is going to be 10 minutes because you guys are waiting on me, even though it still takes you guys 5 minutes to do your job. I'm the weak link. So since you guys are waiting on me and it takes me 10 minutes to do my job, you guys cannot move any faster than the slowest enzyme out there, which is me. So it takes me 10 minutes to do our job, or 10 minutes to do my job. So overall, it's going to take us 10 minutes to make our final product. Now, oftentimes, like I said, it's going to be that first enzyme in that series, in that sequence. And also what we find is once we get enough product built up, that product is going to inhibit that first step. So once we get enough product built up, that product is going to kind of shut me off and tell me not to make any more of A plus B yields AB. And then the rest of the classroom will continue to work until they're finished. And then we use up that product. And once that product has been used up, we have no more product. There's no more product to say, hey, John, stop making your A plus B yields AB. Then I can start up again. And then I'll start the cycle, and then everyone else will start the cycle. And then once we get product again, it's going to shut me off. And then you guys will continue your path, and you guys continue your pathway until we have product, and then the product will shut me off again. And that's an example of a negative feedback loop. All right, just another example of different enzymes for different reactions. You can see a picture version of how the different shapes form into different enzymes. And the product is a rate limiting enzyme, or is the negative feedback loop. All right, factors that increase the rate of enzyme-controlled reactions. Number of enzyme molecules, number of substrate molecules, and the efficiency of the enzyme. Obviously, if we have more enzyme molecules, the reaction is going to happen faster. If we have more substrate molecules, we can build more proteins faster. And the efficiency of the enzyme. Does it take me 10 minutes to work, or does it take me 7 minutes to work? 6 minutes. How efficient am I, or how efficient are you guys? All right, we mentioned phenylketonuria before. Remember, that's our protein deficiency, where that PKU, that amino acid builds up. And we can't eat as much meat as we would like. Actually, we have to eat about 1/7 the amount of protein as a normal male would eat. So you could eat, these are your good foods. Don't eat red meat, chicken, pork, fish, eggs, yogurt, cheese, nuts, legumes, tofu. But basically, it's an accumulation of that enzyme's substrate. We have a missing enzyme that blocks the conversion of phenylalanine, which is an amino acid, into tyrosine. So phenylalanine cannot be converted into tyrosine. So phenylalanine builds up in the blood, and it actually kind of poisons the brain. So eat less protein, eat less phenylalanine. All right, we also have these things called cofactors. Basically, these are non-protein substances that combine with enzymes to help enhance the enzyme activity. Remember that efficiency of the enzyme. That's what coenzymes do. And they're also they're helper molecules that allow enzymes to carry out its function. They're also necessary for some enzymes to function. They help fold enzymes, or those active sites on the enzymes, into their proper conformation or their proper shape. And they also help build or bind enzymes to the substrates. And they can be ions, elements, or small organic molecules, also called coenzymes. So these are going to be like your vitamins and minerals and things that are good for you. So some of your inorganic ions, magnesium, manganese, iron, zinc, cobalt, copper, molybdenum. I can never say that one. Organics, your coenzymes, CoQ10, things that you see on TV. Many coenzymes are derived from vitamins. So, like, for example, vitamin A is used in cellular respiration, vitamin B3, vitamin C, CoQ10. It's an antioxidant and it can actually help alleviate symptoms of congestive heart failure or hypotension. ATP, ADP, which we'll talk about, it's also a coenzyme. Cofactors that alter enzymes. Remember, we talked about denaturation, and that's how proteins become inactive, or enzymes can become inactive due to an irreversible change in that conformation, that secondary or tertiary structure, due to hydrogen bonding or chaperone proteins, and results in that 3D conformational shape. And now that protein and enzyme are unable to bind together due to temperature changes, radiation, electricity, pH changes. All right, so energy is the capability or capacity to change something, or in this case, the ability to do work. We know that energy comes in several forms: heat, light, sound, electrical energy, mechanical energy, chemical energy. Now, if we go back to Newton's first law of thermodynamics, we know that energy cannot be created or destroyed, but it can be changed from one form to another. That's where cellular respiration comes into play. ATP provides energy for cellular work, and most of our metabolic reactions use chemical energy. All of our active processes use chemical energy. So, and that energy is stored in ATP. And it's released, that energy is held in chemical bonds, and it's released when those bonds are broken. So, now we'll talk about that next time. All right, so oxidation oxidation releases energy from glucose via lysis. You really don't need to know that. All right, so in cells, enzymes lower activation energy. We already know that. We're not really going to talk about oxidation. All right, so adenosine triphosphate. So that is ATP. That's the molecule that carries energy in form that the cell can use. It's our main energy carrying molecule in the cell. So, and that energy is used in so many of our cellular processes. So if we look at ATP, we can see that there's, adenosine triphosphate. We can see that there's the adenine molecule, the ribose, sugar, and the 3 phosphate groups. And that energy, let's see, I'm just going to jump ahead in my slides just to see. No. Okay, so what we do, if we remember our equation, ATP goes to ADP. What we're doing is just going to break off this phosphate group and form ADP, adenosine diphosphate, and we're going to have an extra phosphate here. So that's our extra phosphate here, and we're going to release some energy. All right. So when ATP loses that third phosphate or that terminal phosphate group, it becomes ADP. Now we have that double-headed arrow because ADP can be converted back to ATP by putting on that third phosphate group. We'll actually talk about that in a subsequent chapter. That's called phosphorylation, and it requires a little bit of energy to do that. So phosphorylation, it's an anabolic reaction. So ADP can go back into ATP. Remember, 60% heat, 40% energy for cellular repair, stuff like that. All right, so cellular respiration. So remember, this is at the cellular level. And basically, it's to release energy from the nutrient molecules to be used for cellular energy. We see this in four kind of interconnected reactions. There's three reactions that go like reaction 1, 2, and 3. And then, so glycolysis happens, then pyruvate oxidation happens, then citric acid cycle happens. But this electron transport chain kind of happens in simultaneously with the first three. And previously I mentioned that glycolysis, that we have stepwise reactions, and then we have cyclical reactions. So glycolysis and the electron transport chain are stepwise reactions. Step 1, step 2, step 3, step 4, and then once we're done, we're done. Citric acid cycle is a metabolic cycle. It's a cyclical pattern so that once we're done, once we have our final product, as long as we have our final product and as long as we have more source material or source material, we can continue this cyclical pattern until we run out of either product or source material. And actually, I should say glycolysis, pyruvate oxidation, and the electron transport chain are stepwise reaction processes. All right, so we have anaerobic reactions and aerobic reactions. So anaerobic reactions do not require oxygen. They produce much less direct ATP than aerobic respiration. And we'll talk about direct ATP and indirect ATP. Direct ATP is I am producing ATP. Indirect ATP is I am producing something which will eventually lead to ATP. And we'll talk about that later on. Anaerobic reactions take place in the cytoplasm. Anaerobic reactions, glycolysis. Here, because it produces less direct ATP, that energy is going to be transferred to ATP molecules, which we'll talk about later on. That's the electron transport chain. Aerobic reactions require oxygen. They're going to produce more direct ATP than anaerobic respiration. It's going to make the most ATP, both direct and indirect. So let me put that there. Direct and indirect. Takes place in the mitochondria, not the cytoplasm. Pyruvate oxidation, citric acid cycle, and the electron transport chain. All right, so overall process of cellular respiration, we're going to take glucose and oxygen. Really, that's the only two things that are required: glucose, which is sugar, and oxygen. And then we're going to produce carbon dioxide, water, and ATP. That water is actually produced at the end of the electron transport chain. So carbon dioxide, water, and ATP. Now, as we move through the cycles or the steps of cellular respiration, we're going to produce more and more ATP. We're actually going to produce 38 ATP molecules per molecule of glucose. And remember, every time we break down per ATP molecule, if we break down an ATP molecule, energy and heat. All right, so back up here, I told you that glycolysis is going to happen first. Glycolysis is anaerobic, so anaerobic happens in the cytoplasm. Then we have pyruvate oxidation, which is aerobic. It's going to happen in the mitochondria. And then the citric acid cycle is aerobic. It's going to happen in the mitochondria. But the electron transport chain, even though it's aerobic and occurs in the mitochondria, it actually happens kind of simultaneously with all three. So I'm actually going to start with the electron transport chain. So basically, with the electron transport chain, once we kind of do all of our glycolysis, citric acid cycle, and glycolysis, pyruvate, and citric acid cycle, the electron transport chain is the step that's going to complete the breakdown of glucose, and it's going to release carbon dioxide and hydrogen atoms. Also, during glycolysis and citric acid cycle, we're going to produce these things called NADH and FADH2s. And the electron transport chain is going to convert those NADHs and FADH2s to ATPs. And if you remember earlier, I said we produce direct ATPs and indirect ATPs. So direct ATPs is I'm producing ATPs. Indirect ATPs are these things. I'm producing NADHs and FADH2s, and these NADHs and FADH2s are going to go to the electron transport chain, and the electron transport chain is then going to produce the ATP molecules. So whenever we see glycolysis or the citric acid cycle produce an NADH, the electron transport chain will produce 3 molecules of ATP. Whenever we see that FADH2, that's going to go to the electron transport chain, and the electron transport chain will produce 2 molecules of ATP. All right, so we're not going to get into chemistry or upper level chemistry. So I just want you to kind of memorize that NAD plus is a hydrogen carrier, and oxidative phosphorylation consists of the electron transport chain and chemiosmosis. I'm not going to go into why. Just kind of memorize those 2 things. So here is the electron transport chain. If we're not really going to go involved into the electron transport chain, just remember that anytime you see NADH, electron transport chain makes that into 3 molecules of ATP. Anytime you see FADH2, that's going to go to the electron transport chain to produce 2 molecules of ATP. All right, so now we're going to start at the beginning with glycolysis. So Glycolysis. It's a series of 10 reactions. Basically, all we're doing is we're taking glucose and converting it into pyruvic acid. Glucose to pyruvic acid. Remember that glycolysis is air anaerobic, so it does not require oxygen, and anaerobic reactions occur in the cytoplasm or cytosol. And then eventually we're gonna make 2 direct ATPs per molecule of glucose that's broken down. So if we want to look at glycolysis, here's our glycolysis. When I went through AMP, we had to memorize this. We had to memorize glucose, glucose 6-phosphate, fructose 6-phosphate, fructose 1,6-phosphate. We had to memorize hexokinase. We had to memorize all of this. So just be glad that you don't have to. So if we look at our equation, we're going to start with glucose. We're going to end up with 2 pyruvate molecules, 2 NADHs, and 2 ATPs. So here we have 2 direct ATPs right here. And then we also have 2 NADHs. Those NADHs are going to go to the electron transport chain. And remember that every NADH produces 3 ATP molecules from the electron transport chain. So 2 times 3 is 6. So that's how we get 6 ATPs. Now, if we go up here, we have glucose, which is a 6-carbon sugar. Glucose is C6H12O6. And we're going to end up with 2 molecules of pyruvic acid, or pyruvate, whatever you want to call it, which are 3 carbons. And we have 3 of them because we have to balance our chemical equation. So we have 6 carbons over here, and now we have 3 carbons times 2, which is 6. And that's all you're really going to have to remember about glycolysis. So down here, glucose is broken up into pyruvate, but remember, once we break up our glucose molecule in this step, we have to look at, we kind of have to multiply everything times 2 to keep our chemical balance, our equation balanced. So we end up with 2 pyruvate molecules. So if we want to get into it a little bit more, we're actually producing 1, 2, 3, 4 ATPs, but we used 2 ATPs. So 1, 2, 4 minus 2 is 2. So that's how we get this. 4 minus 2 is 2. And then here, our NADH, remember, since we broke up our 6 carbons, we still have to multiply it by 2. We get 2 NADHs, which goes to the electron transport chain. So that multiplies it by 2, or multiplies it by 3. 2 times 3 is 6. So that's how we get our 6 NADHs here. And that's glycolysis. All right, so the next step in is pyruvate oxidation. So remember that glycolysis ends with that 2 molecules of pyruvate or pyruvic acid that occurs in the cytoplasm. And then pyruvic oxidation occurs in mitochondria. It's an aerobic process that requires oxygen in order to do so. So therefore it moves into the mitochondria. So now pyruvic acid is converted into this compound called acetyl-CoA. So we have our 2 molecules of pyruvate get converted into 2 molecules of acetyl-CoA, and we have 2 NADHs. So, and 2 carbon dioxides. So those 2 NADHs go to the electron transport chain. Remember, NADHs, 3 ATPs per NADH, 2 times 3 is 6. So that's how we get our 6 ATPs from our 2 NADHs. 2 times 3 is 6. All right, so from there, we enter the citric acid cycle or the Krebs cycle. So we have our acetyl-CoA combines with this compound called oxaloacetate to produce citric acid. All right, so citric acid, we really don't need to know that citric acids change oxaloacid through a series of reactions. So remember that citric acid is a cyclical pattern. We can actually see that here. It'll keep going around and around and around. So acetyl-CoA actually enters the citric acid cycle, and eventually the final product is oxaloacetate. And it's going to repeat itself if we have enough pyruvic acid or pyruvate and oxygen. Remember, it's an aerobic process. If enough pyruvic acid and oxygen is available. So we start out with our 2 molecules of acetyl-CoA, and we're going to produce carbon dioxide, 6 NADHs, 2 FADH2s, and 2 direct ATPs. So 2 direct ATPs. Then our 6 NADHs go to the electron transport chain. 6 times 3 is 18. Then our FADHs go to the electron transport chain. Remember the electron transport chain, FADHs produce 2 molecules of ATP per FADH2. So 2 times 2 is 4. And then carbon dioxide just goes into the bloodstream, and we exhale that. The only reason why I put this on this slide is because carbon dioxide is produced here as one of the final products. Now, the reason why this statement is on here is because this is a tricky question that's on one of your quiz and tests. So here it says that we produce 4 molecules of carbon dioxide are produced from 2 molecules of acetyl-CoA. Or from 2 molecules of pyruvic acid, because up here we started with 2 molecules of pyruvic acid. Now, I believe the quiz question or the test question says how many molecules of carbon dioxide are produced per molecule of acetyl-CoA or per molecule of pyruvic acid. So you would just divide this by 2, divide that by 2, and you would have 2 CO2 molecules. So just don't get confused when you see that on the quiz or the exam. So per molecule of acetyl-CoA is 2 CO2s that are produced. All right, so that is your citric acid cycle. Here's kind of like an overview. Glycolysis, 6-carbon sugar down to pyruvate occurs in the cytoplasm. We have 2 direct ATPs. Pyruvic acid goes to acetyl-CoA in the mitochondria. Here we have our acetyl-CoA, which enters the citric acid cycle. We produce oxaloacetate to produce 2 direct ATPs plus a bunch of different ATPs through our NADHs and our FADH2s. All right. So, whoops, question for you. After glycolysis and the citric acid cycle, where is most of the energy that was contained in the original glucose molecule now contained? So, another way to read this question is, where do we produce the most of our ATP? Do we produce ATP out of our carbon dioxide molecules? Carbon dioxide doesn't produce ATP. In NADH, FADH2s. Okay, well, yeah, they produce a lot. In the oxaloacetate, that doesn't produce it. Oxygen doesn't produce it. So here, the electron transport chain produces the majority of our ATP molecules. It produces the most energy that was contained in the original glucose molecule. So here we can see that that a total of 34 ATPs were generated from our NADHs and our FADH2s. So going back to glycolysis, we produced 2 ATPs and 6 ATPs from our electron transport chain for a total of 8 ATPs. In our pyruvate oxidation, we produced 6 ATPs from electron transport chain. Citric acid cycle, we produced 2 ATPs, direct 18, so a total of 22 indirect, and for a total of 24. So in total, we actually only produced 4 direct ATPs, 34 indirect ATPs, so a total of 38 ATPs in cellular respiration per molecule of glucose. All right, what substance becomes more abundant during cellular respiration? Glucose, oxygen, ATP, or glycogen? Well, we're breaking down glucose, so that's not going to be the right answer. We're using oxygen, so that's not going to be the right answer. ATP, we're actually making ATP during glucose, or during cellular respiration, so that's going to be the right answer. So no. All right. A cell is unable to break down glucose into pyruvate due to a mutation in a gene that produces an enzyme. What process would this mutated enzyme affect? So, what process converts glucose into pyruvate? Gluconeogenesis, electron transport chain, citric acid cycle, or glycolysis? Well, that's glycolysis. Glycolysis takes glucose and converts it into pyruvate. Which of the following is a product of the citric acid cycle? Pyruvic acid, carbon dioxide, water, or glycogen? Pyruvic acid, that's glycolysis. Carbon dioxide, water. Water is electron transport chain. It's not glycogen. Glycogen is stored glucose. Carbon dioxide. Carbon dioxide is a product of the citric acid cycle. All right, so I said that carbs are our first line of defense for generating energy. So carbs, we know that carbs break down through hydrolysis reaction to form monosaccharides like glucose. And that glucose can enter the cellular respiration to produce energy, carbon dioxide, and water. That's what we just talked about: glycolysis, citric acid cycle, electron transport chain, pyruvate oxidation. All right, but what happens if we eat too much carbs? So carbs can enter the catabolic pathway for energy production, hydrolysis of carbs to monosaccharides. So they enter the anabolic pathway for storage. That's what we just talked about. So, but they can also react to form some amino acids. Remember the chemical structure of carbohydrates. They're CHO. And what's the structure of protein? CHON. So we already have the CH and O. So actually carbohydrates can just get nitrogen from somewhere else and form amino acids. Or if we eat too many carbs, they can get processed and get stored as glycogen. Remember our structure for fats. They're also CHOs. So we can rearrange carbs to form triglycerides. So excess glucose can be converted as glycogen. We call that glycogenesis. Remember, we've talked about this several times now. And then glycogenolysis is how we release that stored glycogen or that stored glucose. But we can also store excess carbs as lipogenesis. That's belly fat, adipose tissue. You eat too many carbs, you get fat. Now, if we look at carbs, we know that carbs can do many different things. Typically, carbs are going to go through glycolysis, citric acid pyruvate oxidation, citric acid cycle, and whatever. But proteins, remember proteins are CHON, they can actually get converted into pyruvic acid and then go through acetyl-CoA and go through the citric acid cycle as well. Fats, remember fats are just glycerol and fatty acids. Glycerol could actually be converted into pyruvic acid. Fatty acids could actually be converted into acetyl-CoA and go through the citric acid cycle. So actually, proteins, carbs, and fats could all get converted and go through the citric acid cycle and be used for energy. And I mentioned before that fats are used for energy use when we sleep or for light exercise. All right, so name the molecule that is common to both carbohydrate and lipid metabolic pathways and connects these pathways metabolically. Glucose, acetyl-CoA, pyruvic acid, or glycogen. So carbohydrate and lipids. So basically, what compound or molecule is common to both of these? What structure is common to both of these? If we look at fatty acids, acetyl-CoA, glycerol, pyruvic acid, carbs, the first one would be pyruvic acid. So fatty acids would get converted into acetyl-CoA. Glycerol could be converted into acetyl-CoA, and carbs can be converted into acetyl-CoA. So that's the answer we're going to go with, acetyl-CoA. All right, Before carbs from the diet can be used for ATP production, what process must occur? Hydrolysis of carbs, hydrolysis of triglycerides, oxidation of glucose, or oxidation of polypeptide chains into amino acids. So we can get rid of some of these statements. So carbs, carbohydrates, amino acids. We can get rid of that one because that's proteins. We can get rid of this one because carbs don't go into fatty acids and glucose. All right, so before carbs from the diet can be used for the production of ATP, what process must occur? hydrolysis of carbs into monosaccharides. So yes, that is an answer. Or oxidation of glucose into glycogen. So that is actually stored glucose. So no, that would not be a viable answer. We actually want glucose. And we know that it's a hydrolysis reaction because we're breaking down carbohydrates into monosaccharides. So A is going to be our answer. All right, DNA. DNA, remember, is deoxyribonucleic acid. It's our genetic material. It stores information on its sequence of nucleotides, our A's, our T's, our G's, and our C's. And that tells our cells how to synthesize or create certain proteins. And proteins are coded for our DNA. They function as enzymes, blood proteins, structural proteins, antibodies, cell membranes. We've already gone over this. And they oversee the four major groups of organic molecules. Remember, carbs, lipids, nucleic acids, and proteins. Remember that genetic information. It's the instructions that tells our cells how to construct proteins. It's stored in our DNA sequence. We have this thing called a genome. It is basically our complete set of genetic information. It's all of our DNA sequences within our nucleus. It contains about 3.2 billion DNA nucleotides. And a portion of that is called our exome. Our exome is a portion of our genome, about 2%, that codes specifically for protein synthesis. And then a gene is a sequence of DNA or nucleotide bases that contains the information about a protein or making a protein. It's our amino acid sequence. It can be thousands or millions of bases long. Remember that genes are packaged into chromosomes. We have about 46 chromosomes or 23 pairs, or we have 46 chromosomes or 23 pairs of chromosomes. And remember that we have gene expression, and it controls which proteins are expressed or produced in each cell type and under what circumstances. It allows the liver cell to be a liver cell, a kidney cell to be a kidney cell. All right, looking at the structure of DNA again. Remember, it's a double helix structure. It consists of two chains of nucleotides. Remember, it resembles that ladder twisted into a spiral. Its backbone is that sugar. The backbone is going to be that sugar phosphate backbone. And then we have that complementary, those strands that are linked together by these hydrogen bonds. So those are going to be our bases. Remember that our A's pair with T's and our G's and pair with C's, adenine with thymine, guanine with cytosine, and vice versa. And that when we look at it, DNA is wrapped around these, we saw this earlier last chapter, DNA is wrapped around those histones. And they form these chromatin structures. So histones are like really, really down here. They're very microscopic. All right, remember, A pairs with T's, G's pair with C's. All right, so it says that the nucleotide chains are anti-parallel. They point in opposite directions. So think of like you have opposite sides of the street. All the cars are facing this way on this side of the street, and all the cars are facing this way on this side of the street. So they're all facing in opposite directions on both sides of the street. So here you have this O that's pointing up. You have this O that's pointing up. You have this O that points down. You have this O that's pointing down. So that's just what anti-parallel means. The backbone is pointing in opposite directions. They're anti-parallel. Again, remember A's and T's go together, G's and C's go together. And I guess I block this out, but these are purines, and these are pyrimidines. I guess I deleted the purine portion of the title. So purines have these 2-ring structures, and pyrimidines only have 1-ring structures. Remember, uracil is only for RNA. DNA has thiamine instead of uracil. So pyrimidines are single ring structures. Purines are 2-ring structures. So that's why we have A's with T's and G's with C's. A purine will always bind with a pyrimidine. You will never have a purine with a purine. You'll never have a pyrimidine with a pyrimidine. So you'll always have a 2-ring with a 1-ring structure. And that's just to keep the DNA approximately equal in width. You don't want to have a 1 ring with a 1 ring, then a 2 ring with a 2 ring, then 1 ring with 1 ring, 2 ring with a 2 ring. You want the DNA approximately the same width to keep that ladder shape when it's coiled up. All right, so RNA is different than DNA. We know that it's single-stranded. It still has that sugar, but in this time, instead of deoxyribose, it's ribose, hence the name ribonucleic acid. We know that it's uracil instead of thiamine. The other 3 bases remain the same, adenine, guanine, and cytosine. RNA is going to be shorter than DNA. We have 3 different types of RNA: messenger, transfer, and ribosomal RNA. Here, I would probably just look at this one. I have a hard time remembering which ones do which. Structure of RNA, it doesn't coil up in that helical structure like DNA does. All right, so Innocence Project. So remember that the genome consists of about 3.2 billion DNA bases. The Innocence Project basically looks at old cases and tries to free innocent criminals that have been wrongly convicted due to now DNA profiling. And actually, several hundred prisoners have been freed due to the backlog of DNA testing, or, back in the olden days, you had a black prisoner who was convicted from an all-white jury. We didn't have DNA testing back then, and a lot of racial bias, and now this Innocence Project is now trying to free people who were wrongly convicted. All right, so we talked last chapter about DNA replication, and that happens during interphase, and that during mitosis, hopefully we produce a daughter cell that is the exact copy of the original cell. And it's obviously necessary so that the cells have a complete set of genetic information. So what happens in DNA replication is that the hydrogen bonds between those base pairs, the A's, the T's, the G's, and the C's, those are going to break up. So your secondary structure, hydrogen bonding, will break up. The DNA strands are going to unwind and separate. Then those new nucleotides are going to pair up with the exposed bases. With the original A will pair up with a new T. We have this enzyme called DNA polymerase. And then we have other enzymes that will connect this new sugar backbone. But eventually, new nucleotides are obviously going to be assembled into that DNA strand that is complementary to the original DNA strand. So you can see here that we have the hydrogen bonds here. Eventually, they're broken up, and the space gets wider, farther apart, and we have a new G that comes in, or no, oh no, that's falling off. Never mind. Um, that's falling off. Don't. No, I'm looking at the wrong thing. Um, on this side, I'm sorry. I'm looking at the wrong thing. So what I was looking at, this is genetics. We're not even going to go into this. There's actually a 3' end and a 5' end of DNA, which you'll learn about in genetics. Um, so um, this so DNA kind of remember how we have that anti-parallel structure of DNA. Um, so we have a 3' end and a 5' end. So when we replicate DNA, DNA will, on one side of the strand, DNA will start replication at the top, and on one side, DNA will start replication at the bottom. So here you can see that on the right side that the DNA has already replicated, kind of starting from the top to the bottom. So this T has already paired up with an A. Here it's already paired up. Here it's already paired up. Here it's already paired up. Now the C is pairing up with the G, and the C is coming to pair up with the T, and eventually the T will come pair up with the A. And then once it's done, then on the right side, then it'll jump back up here and start pairing up here. And then on the left side, you can see that it's going to build up from the bottom up. So this G is going with the C. This C is going with the G. So I was getting my 3' and my 5' in mixed up. So anyway, like I said, we're not going into that. All right, but what we are going into is how to do DNA replication. Remember, DNA replicates during interphase inside the nucleus. All right, so all we're doing is we're going to break up the DNA strand and produce a complement. You should already know how to do this. Anytime you see an A, put a T. Anytime you see a G, put a C, and vice versa. So an A would become a T. T would become an A G to a C, T, A, T, A, G, C. And I broke these up into groups of 3. 1 for more visual ease, and 2, you'll see why later on we have these things called codons and anticodons, which we'll get into later. C to G, C to G, A to T, and so on. So I hope this is an easy one. This shouldn't be anything new to you. All right, so protein synthesis. We have that sequence of DNA, of 3 DNA nucleotides, that's going to provide that template for the complementary RNA. So we've done our DNA sequence, and we've made a complementary DNA sequence. Now we're going to make RNA from that DNA. And we have 2 terms that we're going to use called transcription and translation. So we're going to take that DNA sequence and make an RNA sequence through transcription and translation. And we're going to take that, the 3 nucleotides, and it's going to represent either an amino acid or signals to start or stop that synthesis of whatever protein we're going to create. We have 20 amino acids in our body, but we have to know when to start and stop this whole replication process. And of course, we have to have enzymes that catalyze both this transcription and translation process. All right, so transcription is basically copying DNA and producing mRNA, which is messenger RNA. So we're going to use that DNA as that copy of the master code. We use that as a template. This still occurs in the nucleus. And we're going to use this enzyme called RNA polymerase that's going to catalyze the formation of messenger RNA. So what's going to happen is that RNA polymerase is going to somehow go along that DNA strand, and it's going to eventually say, oh, I found this promoter. It's going to bind to a promoter. And a promoter is just a region of DNA sequences, a bunch of bases that begins a gene. It's about 100 to 1,000 base pairs long. And it says, okay, this is where I'm about to begin my protein creation or my protein synthesis. So it's going to unwind that DNA to expose that gene to code for some protein, whatever protein we're trying to create. So it's going to use this mRNA nucleotide pair with DNA bases. We're going to take that DNA that we just did and create a messenger RNA strand. It says it uses codons. And we'll talk about codons later on. But again, we have to know when to start and when to stop. And there are 3 stop codons. And I'll tell you right now, there's 1 start codon and 3 stop codons. Once we've started, once we've stopped, creating our messenger RNA strand, that messenger RNA strand's released, and then that DNA strand kind of winds back up into that double helix. That mRNA strand leaves through the nuclear pore, attaches to the ribosome in the cytoplasm. That ribosome will go to the rough endoplasmic reticulum, Golgi apparatus, get packaged into vesicles, and possibly leave the cell for wherever it needs to go to. All right, so when we go from DNA to RNA, remember that we don't have any more T's on the RNA side. That A is going to pair with a uracil. All right, so we have our DNA. We've replicated our DNA. So now let's do our transcription. So we're going to make our mRNA. So anytime we see a T, we're going to make an A. So now we have an A. Instead of a T, we're going to make our U, uracil. C to G, A to U, A to U, C to G, G to C, so on. So the process is very similar except for our A's to U's. And that's just transcription. We've taken our DNA and we've made an mRNA strand out of our DNA. All right, so now translation. It's basically a protein synthesis. What we're doing now is we're going to produce a sequence of amino acids. We're going to take that genetic information within the mRNA strand and build a polypeptide chain. We're going to build a protein chain. We're going to take these amino acids and join them by polypeptides. But As of right now, all we have are A's and U's and G's and C's. We don't have any amino acids. We need to come up with amino acids. So remember that that mRNA strand is in the nucleus, and that mRNA strand leaves the nucleus, and it's going to then go to the ribosome in the cytoplasm, and that's where we're going to see protein synthesis. And that's when we're going to determine the amino acid sequence. So what we're going to do is we're going to have this thing called transfer RNA. It's going to carry and bind amino acids along that mRNA strand on the ribosome. It binds amino acids, transports it to the ribosome, binds to the mRNA strand according to its sequence, and it adds the amino acids to the growing polypeptide chain. So each tRNA transfer RNA contains a sequence of 3 nucleotide bases, anticodon, which binds to the blah, 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 blah. All right. And then remember, and I'm just going to say this now. So far right now, I know that this is confusing. We'll get to what this means here in a second. Remember that chaperone proteins assist in that 3-dimensional conformational shape, that tertiary structure of a protein. So if that protein is produced with the correct amino acid sequence, but does not have the correct 3-dimensional shape, then that chaperone protein associated with a protein folding is probably not functional. So remember, we have to have functioning chaperone proteins. All right, so I said that we have 20 amino acids. So with that, we have these things called codons. And that's why I kind of grouped all of these in groups of 3. A group of 3 is called a codon. And we have 4, an A, a U, and a G, and a C can be here. An A, a U, a G, and a C can be here. And an A, a U, and a G, and a C can be here. So we have 4 different combinations for the first one, 4 different combinations for the second letter, 4 different combinations for the third letter. So 4 times 4 times 4 is 64. So we really have 64 possible codons, or 64 possible 3-base sequences on our messenger RNA. But what we find is that AUG corresponds to the amino acid methionine, and that's always going to be our initiation codon or our start codon. And remember that I said we have 3 stop codons, and that's going to signal the end of your protein synthesis. So really we only have now 61 codons for those 20 amino acids. So here are our codons. So here we have AUG. That's going to be our start codon. Remember I said that's methionine. We have 3 different stop codons over here. And you can see if we have, CUU, that's going to correspond to leucine. You can see that all of these correspond to leucine. Actually, these 6 correspond to leucine. All of these correspond to leucine. These 4 correspond to the amino acid serine. These 4 correspond to the amino acid proline. These 2 correspond to the amino acid phenylalanine. So you can see that whatever codon or whatever group of 3 that we have corresponds to an amino acid. All right, so how many amino acids can be encoded from a sequence of 333 nucleotides? So if we have 333 nucleotides, so let's say we have, so that's 333 bases. So we have 333 letters. So now we're going to group those letters into groups of 3. So 333 divided by 3 is 111. So A is going to be your correct answer. Now, what would occur if a DNA sequence for an enzyme lacked a termination signal? Translation would not have an endpoint. The incorrect amino acid would be incorporated into that final enzyme at that point. Transcription would not have an endpoint, or DNA replication would not be possible. So if we don't have a termination signal, basically it's going to go on forever and ever. So basically, we're not talking about DNA replication because we've already replicated our DNA. We wouldn't put in the wrong amino acid. So now are we talking about translation or transcription? Basically, it would be transcription because that's when we would find our endpoint, when we would bring down that final stop codon. All right, so we've done our transcription. We've already done this. So what we would need to do is look at this and kind of find out what those codons correspond to. So now let's look at AUG. AUG is our start codon. So there's our start codon. UUG we would transcribe that as, so this would be our transcription. Gets converted into AAC. AAC is asparagine. So transcription would bring down the amino acid asparagine. CCA would get transcribed as GGU. GGU is glycine. GGU is glycine. And then CGU would be GCA, which is alanine. And then UGA is our stop codon. So our amino acid sequence, or our polypeptide chain, would be asparagine, glycine, alanine. All right, this is just a picture version of what's going on. Out of another picture. All right, what is the function of RNA polymerase? I gave you the definition several slides ago. It's going to be C. It assists in the transcription process of a DNA molecule to messenger RNA. All right. What happens here? A, went to T, T to A, G to C, C to G, T to G. Oh, T should go to A, not to G. So the cell correctly copied. Nope. An error occurred during transcription. No, because we're doing a DNA sequence. So transcription is during RNA. There was an error during DNA replication. Yes. An error occurred during translation. No. So C would be your best answer. All right, changes in genetic information. So about 99.9% of the human genome is shared among all people. So there's really no, or among all humans. So there, you really can't tell you from me just by looking at our DNA. No inferences can be made about similarities or differences between two individuals really just by looking at our DNA. About only 0.1% of our genome varies among people that affects our health, appearance, and really has no observable side effects or observable effects. We do have mutations in or errors in the DNA sequence, and that occurs when bases are added, changed, or deleted. And this could result in disease because the encoded protein has a different function than the original. And, we saw that, here, GGG is very different than GAG, which is what it should be. So GGG, let's see, GGG is glycine, and GAG would be glutamic acid. So that would be because the encoded protein has a different function than the original. But that's actually not a mutation. Because a base was not changed, added, or deleted. That's an error in replication. So that's an error versus a something being changed, added, or deleted. All right, so error mutations could be an extra nucleotide is inserted into the DNA sequence. Something is deleted, a 10 base length of DNA is deleted during replication, or cytosine is replaced with adenine. All right. Or it could be induced due to a mutagen. Ultraviolet light, we know, causes skin cancer. Nitrous acid can convert cytosine to uracil. X-rays can break down bonds in DNA molecules. We talked about different types of radiation. Ethidium bromide is a chemical that directly binds DNA and deforms that double helical structure. But if a tRNA brings down the wrong amino acid during translation, that is just an error, not a mutation. Now, we'll get to that in a second. So here, sickle cell disease. We have a T that codes for glutamic acid, but if we brought down an A instead, a mutation, that could be a mutation because we brought down amino acid, or we brought down valine instead of glutamic acid. Sometimes mutations are harmful. In this case, it causes sickle cell disease. In some instances, mutations are not harmful and do not affect health. because let's say that we were supposed to bring down, let's say we were supposed to code for CCU, but instead we coded for CCC. Well, we still brought down proline. We still brought down the same amino acid. So we didn't, that mutation did not lead to a change in the amino acid sequence. So that mutation was not harmful. All right, many mutations do affect health. We just saw the sickle cell disease example. It causes abnormal hemoglobin structure, causes that red blood cell to kind of sickle up, and these red blood cells can get caught up in the capillaries and clump together in low oxygen conditions, and it can be very painful to the patient. Now, rarely mutations can provide an advantage to health. One example is that we see this in some Scandinavian people, that people who have been exposed to HIV, the virus does not attack the person's immune system because that receptor that the virus would attach to the white blood cell, the T cell, is incomplete. So the virus does not get into the T cell and therefore doesn't cause havoc on the person's immune system, kill off their immune system. All right, so how do we protect against mutation? We have this DNA damage response. We can possibly correct that nucleotide or that mismatched nucleotide. We can go in and kind of splice out the incorrect portion and reinsert the correct portion. We can kind of scrap that whole protein to get all together if we can, if the body wants to, or we can kind of kill off that cell if the body wants to do that. We also have 2 copies of the chromosome. If the body if the other cell is functioning, then the body can kill off the mutated cell, so long as the other cell is normally functioning. Remember that the spindle fibers pull the chromosomes apart, and if one cell is good and the other cell is bad, we can go with the good cell. So, all right, so that is the end of chapter 4. So let me know if you guys have any questions. I know there was a lot covered in this chapter. So I'll see you next chapter. All right, take care.